Assalamu alaikum. So today we will study the structural and terminal units of salivary glands, in which we will discuss the ductal system of the salivary gland and the individual cells of the salivary gland, right? The salivary gland, as you all know, they are compound tubulo-acinar merocrine glands. Merocrine means they pour their secretion with no cytoplasm or cell content is lost during the secretion of the cells, right? And they are tubulo-acinar as they are tubule presence, and they are compound glands. Compound means the multiple ducts are present in it, ठीक है? And it is just like when you saw a tanky. टैंकी देखिए ना आप लोगों ने, ऊपर सबसे ऊपर लगी होती है छत के, वहां से पानी डिफरेंट पाइप से होकर नीचे जाता है और बकेट में जाके जमा होता है. बकेट इज योर ओरल कैविटी, ठीक है? और इसको डिफरेंट जो है डक्ट्स ने ज्वाइन किया होता है, ठीक है? और वहां से ये ट्रैवल करके सीधा सीधा जाकर उस ओरल कैविटी में जमा हो जाता है, ठीक है? सो द एग्जाम्पल यू कैन सी, you can correlate it with this tanky, and you can correlate it with the bunch of grapes. bunch of grapes देखे ना आप लोगों ने, जिसमें stems और branches लगी होती हैं. ducts are branches, ठीक है? So these, it is very easy to understand, and the secretions of different cells are different in the salivary glands, right? So we will proceed. The salivary gland, its basic structural and functional unit is SNR, right? They are the cells which is present in the salivary glands. Epithelial secretory unit is composed of serous and mucous acini along with myoepithelial cells. जो सलाइवरी ग्लैंड्स में जो टर्मिनल यूनिट होता है एपिथेलियल, जहाँ से सलाइवा सेक्रीट होता है, दैट इज कंपोज्ड ऑफ सिरस और म्यूकस एसिनाई, जो कि सिरस के म्यूकस सेक्रीशन जो है वो सलाइवरी ग्लैंड्स में रिलीज करते हैं, राइट? एंड दीज सेल्स लाई ऑन द बेसमेंट मेम्ब्रेन, and there is separation between the underlying mesenchymal cells and this basement membrane, right? The central lumen which is present between these cells is star shaped because of the extension between the cells, right? Now we will see the ductal system. In the ductal system, it is similar to this bunch of grapes. Have you ever seen the bunch of grapes? There is the main duct. This is the main duct जो कि स्टेम होता है ग्रेप्स में. इफ यू इमेजिन ग्रेप्स, ठीक है, उसमें फिर साथ यहाँ पर ब्रांचेस दी होती हैं, ठीक है? एंड these branches are present in the salivary glands. और यहाँ पर ये terminal unit है, that is the secretory terminal unit in which these canaliculi is present. The ducts between these secretory units are the intercalated ducts, right? And this duct, basically the function of this duct is for the passage of saliva, right? And this is the main excretory duct which releases the saliva into the oral cavity, right? जो हमने different ducts पढ़ी, जैसे कि parotid gland में Stenson's थी, Wharton's थी, these are the main excretory ducts, ठीक है? And this is the striated duct in which the striations are present, and the reabsorption of ions and exchange of ions takes place in this striated duct, right? And the saliva, as you know, the saliva is watery. Basically the 99.9% of the water is it contains the 99.9% of saliva contains water, ठीक है? सिर्फ 0.1% उसमें जो है बाकी ingredients present होते हैं, जैसे कि कैल्शियम हो गया, immunoglobulins हो गए, हमारी एंटीबॉडीज हो गई, देयर इज बफरिंग एक्शन जो है pH लेवल की वजह से, एंजाइम्स हो गए that are present in the saliva, that all gather up together only in the 0.1% of the saliva, ठीक है? कुछ बुक्स में जो है 0.1 to 0.5% की रेंज दी है, और कुछ में 99.5% लिखा है. The variations are present in the books, ठीक है? Now this diagram shows the histology of the salivary glands very clearly. Here we can see the secretory end units. These are the secretory end unit containing the serous acini or the mucous acini. These are the serous acini and these are the mucous acini. These cells along with this myoepithelial covering make the secretory end unit, yet terminal unit of the salivary glands, right? These serous and mucous acini lie on the basement membrane, and uh, they have these junctions. These serous and mucous acini are joined together by the epithelial junctions like desmosomal junctions. Zonular occludents can be present between these cells, and these cells carry the intercellular canaliculi, which makes a central lumen a bit star shaped. Okay? And these shape of the cells make the end piece roughly spherical or tubulo-acinar. Basically, that the salivary glands are tubular exocrine glands. Okay? They pour their secretion through ducts, and they have merocrine secretion. Merocrine secretions are those secretions in which the cells pour their secretion through exocytosis into the central lumen, and then from here they travel towards the main duct. The mucous acini has got a larger lumen as compared to the serous acini. The serous acini contains 8 to 12 serous acini are normally present in a secretory unit, whereas the mucous acini are greater in number. They may be 12 to 24 in a secretory unit, and there are this intracellular canaliculi is not present in the mucous cells, whereas if they could be present when they have a serous demilunes. So these are the serous demilunes which is present over these mucous cells. Then the intercellular canaliculi can be present, and these are the intercalated ducts which is present between these secretory units, and it has got cuboidal cells lining cuboidal epithelial cells lining on the basement membrane, right? These are the striated ducts. You can see the striations over here, right? These striations are present for the absorption and reabsorption of for the secretion and reabsorption of ions takes place in this striated duct. And here the this is the main reabsorption duct, and the more energy is needed over here, and the cells becomes very rich in its organelle, and the reabsorption and secretion of ions takes place here, right? And this is the main excretory duct which opens into the oral cavity and releases the saliva after the modification, after its ionic modification, right? The terminal secretory unit of the salivary gland is made up of epithelial secretory cells that could be serous or mucous. ठीक है? These serous and mucous cells along with myoepithelial covering, these are the myoepithelial cells, as you can see. These are the contractile cells. Myoepithelial cells are contractile cells which with its contraction and relaxation release the saliva into this lumen, and it is then poured through intercalated ducts into this main excretory duct, right? These cells, mucous and serous cells, are present in the secretory unit of SNI. These serous cells are pyramidal in shape, having spherical nucleus near the basement membrane, and this basement membrane is separated from the underlying mesenchymal cells. In the mucous cells, the nucleus is flattened and is more basally present as compared to the serous cells, right? The serous cells produce watery secretion, and it has high protein to carbohydrate ratio, whereas mucous cells secrete the secretion in which there is protein की रेशो कम होती है और कार्बोहाइड्रेट की ज़्यादा होती है, while in serous cells the protein ratio is higher than the carbohydrate, right? So these cells, the serous cells, contains different rich protein synthesizing organelle, and they are 8 to 12 in an SNS, normally 8 to 12 in SNS, whereas mucous cells are higher in number, and the serous cells are connected with each other with intercellular junctions. The intercellular junctions could be the zanula occludens, which is a tight junction. Their junctions could be intermediate, like zonula adherent, and more desmosomal junction like maculae adherent could be present in between them, right? These junctions control the permeability of the cells. The central lumen of the cells via different fine ducts, they grow larger and larger and pour its secretion to the main ducts. ठीक है, different ducts जो हैं they join together and makes a larger lumen and pour its secretion towards the excretory duct. ठीक है, जो ये intercalated ducts है इनका lumen थोड़ा कम होता है, फिर ये एक साथ join करते हैं, फिर lumen थोड़ा ज़्यादा हो जाता है, और फिर ये जो मेन एक्सक्रीटरी डक्ट के जरिए जो है द ओरल कैविटी में जो है वो सलाइवा सिक्रीट होता है. स्ट्राइएशंस ये आप देख रहे हैं, दिस इज द स्ट्राइएटेड डक्ट जिसमें स्ट्राइएशंस हैं, और परमेबिलिटी जो आयंस की मॉडिफिकेशन है वो स्ट्राइएटेड डक्ट्स में होती है. ठीक है, यहाँ पर आयंस एक्सचेंज होते हैं और मॉडिफिकेशन होती है सलाइवा की, और फिर वो जाकर मेन एक्सक्रीटरी डक्ट में चले जाते हैं, राइट? अच्छा, जो ये म्यूकस सेल्स हैं इसमें इंटरसेलुलर जंक्शंस प्रेजेंट नहीं होते, वेयर एज सेरस सेल्स में जो है इंटरसेलुलर जंक्शंस प्रेजेंट होते हैं. द इंटरसेलुलर जंक्शन इन दिस म्यूकस, इसमें डिफरेंट इंट्रासेलुलर जंक्शंस प्रेजेंट होंगे, लेकिन इंटरसेलुलर जंक्शंस बहुत कम प्रेजेंट होते हैं. अगर इंटरसेलुलर जंक्शंस वहाँ प्रेजेंट होंगे जहाँ पर यहाँ पर सेरस डेमिल्यून्स प्रेजेंट होते हैं. सेरस डेमिल्यून्स और इन म्यूकस सेल्स के दरमियान जो है इंटरसेलुलर जंक्शंस प्रेजेंट होते हैं. आपको लास्ट पिक में शायद सेरस डेमिल्यून्स नज़र आ रहे होंगे, इसमें इतने ऑब्वियस नहीं है. ठीक है, और जो सेरस और म्यूकस सेल्स हैं वो ज़्यादातर मिक्स्ड सेक्रीशन में जो है ये प्रेजेंट होते हैं, जैसे कि सब मैंडिबुलर ग्लैंड्स हैं उसमें ये सेरस डेमिल्यून्स जो हैं वो प्रेजेंट होंगे, राइट? अच्छा, जो सलाइवा की जो कॉम्पोजिशन है कहीं पर 99% भी लिखी है, कहीं पर 99.5% भी लिखी है, और कहीं पर 99.9% भी लिखी है. सो जो आपकी ऑर्गन्स में जो है वो 99% लिखी हुई है. ठीक है, सो यू कैन गिव द रेफरेंस ऑफ द डिफरेंट बुक्स. सो द रेंज इज प्रेजेंट 99 to 99.9% जो है वो सलाइवा में वाटर प्रेजेंट होता है. ओके, so now we will study the individual histology and function of the serous cells and mucous cells. This is a picture of serous cells. You can see the nucleus which is rounded and near the basement membrane. ठीक है, and this is the apex of the serous cells. These serous cells show accumulation of secretory granules. You can see there are numerous rough endoplasmic reticulum present which just lateral to this nucleus, and you can see the granules are present in the cytoplasm of this serous cells. These granules are zymogen granules which is present in the serous cells, and these zymogen granules is one of the characteristics of these serous cells, as these granules are not present in the mucous cells, and they travel and they are made in these ribosomes, and they travel toward the lumen of this serous cells and are secreted from this vacuole by exocytosis. These granules are produced by glycolated proteins which are released into the vacuole. The number of secretory vacuoles differ on the basis of activation of the cells. When the cell is activated and they produce these granules, the produce and release these granules, the number of granules will be less in the cells, as many of them are released in the vacuole, right? In the serous secretion, the protein ratio is more than the carbohydrate, while in mucous cells, the carbohydrate ratio is higher to protein, right? And these serous cells have got the enzymatic activity like for acid phosphatases, esterases, and glucuronidase enzymes are present in these serous cells, right? The serous cells show an active synthesis of protein, and it is rich in protein synthesis organelle. As you can see, there are abundance of rough endoplasmic reticulum and mitochondria, and the Golgi complex are present more in number, as it is a protein secreting cell, so it needs all the protein synthesizes organelle in its cytoplasm. There are intercellular junctions present between these cells, which could be, as I have told you, junctional complexes like desmosomal or zonulo-occludent for tight junctions could be present. These junctions basically control the permeability of the cells, right? The ribosomes and the rough endoplasmic reticulum plays a vital role in the synthesis of protein. They are basically nucleus mein, as you have been studied long for in the biology and in your anatomy, that the ribosomes are of three types: messenger RNA. ठीक है, ribosomes are basically composed of ribonucleic acid and proteins. There are messenger RNA, transfer RNA, and the other ribosomal RNA, I guess. ठीक है, so ये इसमें क्या होता है कि द न्यूक्लियस में जो मैसेंजर RNA है वो इनकोडेड सिग्नल लेके जाता है टूवर्ड द साइटोप्लाज्म, जहाँ पे there is signaling protein present which is recognized as this rough endoplasmic reticulum, and they encode this message of synthesis of protein. So the ribosomes, the messenger RNA takes an encoded message which is translated by ribosomes, and an appropriate protein is made of 10 to 15 amino acids, and this is called as a signal sequence, right? And after that, this signal sequence attaches to the membrane of the rough endoplasmic reticulum. This is the rough endoplasmic reticulum. This is recognized by by an enzyme, and it detaches the protein over here. So it is recognized by this rough endoplasmic reticulum, and the signal sequence is removed, and the protein is synthesized, and it is sent to the cisternal space of the rough endoplasmic reticulum. I hope you all know what is the cisternal space. Cisternal space is basically the inside space of these folds of these rough endoplasmic reticulum, and from here it travels to the Golgi apparatus. Golgi apparatus is connected to this rough endoplasmic reticulum by budding vesicles, and these budding vesicles, इसमें इतने पिक्चर में क्लियर नहीं है budding vesicles. I wish कि मैं आपको उस तरह से दिखा सकती. बडिंग वेसिकल्स के साथ ये जब कनेक्ट होता है, फिर वो प्रोटीन वहाँ से ट्रैवल करके वैक्यूल्स में चला जाता है. ठीक है, देयर आर सर्टेन एंजाइमेटिक एक्शन विच इज गोइंग ऑन in such time in this time. बडिंग वेसिकल्स के बाद ये वैक्यूल्स में चले जाते हैं, ग्रेन्यूल्स, और ग्रेन्यूल्स उस वैक्यूल से पास होकर दे empty into the lumen by the process of exocytosis. ठीक है, so these are the different stages. It is a complex process of the formation of the granules and the transfers of these granules up to the lumen of the cells. ठीक है. And there is the granules are formed. The granules, when are formed, they are first in premature stage, then the mature stage, and then they pass towards the lumen of the cells. ठीक है. The most common modification which takes place in this process, and the important one as well, is the addition of the carbohydrate to these proteins, and this process is known as glycosylation. The glycosylation starts at the rough endoplasmic reticulum and ends in the Golgi apparatus, and from here it travels. The glycosylation takes place, and the zymogen granules are glycosylated, and they are released into the lumen of the cell, right? And the serous cells devote 80% of its energy in production of these secretory granules. And एक्सोसाइटोसिस का शायद मैंने बताया कि नहीं. एक्सोसाइटोसिस में ये होता है कि वैक्यूल इस मेम्ब्रेन के साथ फ्यूज कर जाता है. फ्यूज जब करता है तो इसमें से सिर्फ ये ग्रेन्यूल्स जो यहाँ पे बने होते हैं, द जाइमोजन ग्रेन्यूल्स, वो सिर्फ रिलीज होते हैं. देयर इज नो लॉस ऑफ साइटोप्लाज्म और द सेल ऑर्गेनेली इन दिस प्रोसेस, राइट. वो आराम से बस ग्रेन्यूल्स जो हैं वो ल्यूमेन में रिलीज कर देता है. ठीक है. एंड देयर आर अदर एक्टिविटीज आर आल्सो गोइंग ऑन, लाइक द smooth endoplasmic reticulum. They also synthesize the intracellular protein which is present. The mitochondria provides the power to the cell to undergo the process of protein synthesis, and the lysosomal and the enzymatic activity is also going on. As I have told you, the enzymes are present which decode the signal, or it also take part in the destruction of any foreign material. So these all cellular activities are going on beside the production of these zymogen granules. I hope you all have understood the activity of serous cells. It is very well detailed, very well and written in the book Organs. So you guys go for Organs. उसमें भी लिखा हुआ है 10 gates में, आप 10 gates भी देख सकते हो. मैंने Organs से पढ़ा है तो आप Organs को भी टैली कर सकते हो. ठीक है. So this is another very good pic of serous cell, in which you can see the different, you can see the spherical nucleus and all. From here the messenger RNA travel towards the rough endoplasmic reticulum, and from here it travels towards the Golgi apparatus, and from the Golgi apparatus the granules travel toward the lysosome, heterogeneous electron, as it is labeled, the secretory granules is present over here, and from here thus vacuole transfuse with this membrane and release these granules into the lumen, right. So this was the process of the production of zymogen granules, and the other activities also going on, like the जो आप लोगों ने पढ़ा है क्या कहते हैं उसे, Krebs cycle, and other cellular activities also going on. The smooth endoplasmic reticulum, intracellular secretory protein बनाते हैं. The junctions are present. You can see these junctions are present, and there is this was about the mechanism of the zymogen granules, which I have repeated in this diagram, the histological diagram. I find it more good, तो मैंने इसको repeat कर दिया है. So I hope all of you get it. So please if you guys study करें इसको थोड़ा सा अपने organs में जाके, और थोड़ा सा इसमें मेन चीज़ें जो हैं. ठीक है. serous cells में glycosylation किसे कहते हैं. ठीक है. First question is, what is glycosylation? ठीक है. What is the carbohydrate protein ratio in the serous cells? ठीक है. And the shape of the serous cells, आप उसको अच्छी सी एक cellular picture बनाएं serous cell की, उसमें ये सारे organelle और ये सब कुछ आप लोगों ने लेबल किया हो. ठीक है. जिस तरह इस पिक्चर में लेबल किया हुआ है, लेकिन ये जो पिक्चर आप बनाएंगे ये आपकी हैंडमेड पिक होनी चाहिए. ठीक है. यू डोंट हैव टू टेक इट फ्रॉम द बुक. You have to draw it yourself and you have to send me the pic of the serous cell. ठीक है, handmade serous cell बनाना है. और आपने इसकी ग्लाइकोसाइलेशन, व्हाट इज ग्लाइकोसाइलेशन नंबर 2. ठीक है, ये सारे 3 मार्क्स के हैं. ठीक है, और इसके अलावा आपने लिखना है what is the salivary gland, the proper definition. tubuloacinar जो है कंपाउंड ग्लैंड्स हैं. ठीक है, एंड द picture of the structure of the salivary glands, जिसमें आपने शो करने हैं canaliculi, intracellular canaliculi, intercalated ducts, the main excretory duct. ठीक है, एंड द सेक्रेटरी एंड पीसेस जिसमें सेरस और म्यूकस सेल्स शो की हो. तो 2 आपने पिक्चर्स बनानी है, सेरस सेल की और सलाइवरी ग्लैंड के डक्टल सिस्टम की. ठीक है, और इसके अलावा आपने लिखना है व्हाट इज द प्रोसेस ऑफ ग्लाइकोसाइलेशन. दिस इज योर असाइनमेंट एंड आई होप यू विल डू इट एंड डू योर बेस्ट. आई एम वेरी हैप्पी जब मैं देखती हूँ कि स्टूडेंट्स ने बहुत अच्छा लिखा होता है, और मुझे बड़ी खुशी होती है कि स्टूडेंट्स अच्छा करके भेजें, वो उस चीज़ को समझें और उस पर कोशिश करें और फिर मुझे भेजें. जो कॉपी पेस्ट करते हैं उनसे खुद को पता है कि क्या है वो क्या नहीं है, लेकिन द थिंग इज़ कि मुझे बड़ी खुशी होती है जब मैं कुछ स्टूडेंट्स से बहुत अच्छा करके भेजते हैं, मैं उनको फिर एक्सेलेंट करें मार्क भी देती हूँ. तो आप लोग जो है जो भी काम करता है, आई रियली अप्रिशिएट दैट. तो आप लोगों ने हैंडमेड पिक्चर्स बनानी है इस दफ़ा, आपने उससे नहीं, वेब से नहीं करनी. ठीक है, और आई होप यू ऑल गेट इट एंड सी यू इन द नेक्स्ट क्लास. So I hope you all are getting my lecture as salivary gland I am teaching for the first time. I hope you all are getting it. ठीक है.